Okay, well, thanks very much to the organisers for the opportunity to talk today. I'm going to talk about a tool that we've been developing at TGAC on and off really for the last couple of years called MetaCortex, which is a development of the Cortex assembler sort of aimed at metagenomic data. And then I'll talk about a particular viral project we've applied it to. For those of you that are not aware of TGAC, we're a relatively new institute in Norwich, about an hour, just over an hour, sort of northeast of Cambridge, and we're in the Norwich Research Park, which is this whole site here. This is the TGAC building, and we're kind of right next to the John Innes Institute, if you've come across John Innes, the John Innes Centre rather. And we're about 4 years old. We've got quite a lot of sequencing, but we've got quite a lot of bioinformatics and computational biology, and so more than half of the institute is doing bioinformatics and computational biology. So the talk then is sort of really three parts. I wanted to talk about the challenge of assembly in general. So I guess quite a lot of this will be kind of revision on genomic assembly and why genomic assembly approaches don't work with metagenomic data. Going to talk about one answer in inverted commas, because we've sort of heard a lot about how difficult assembly is. So it's not an answer, but it's maybe a step on the journey. And then talk about these cuddly creatures here, which are straw-coloured fruit bats, and a project we looked at with trying to understand the virome of these bats. So, I mean, why do we want to do metagenomic assembly? Well, sort of simple answer is we want longer sequences. But I mean, it has the potential to help us with improved accuracy for binning and classification, to help us to discover novel organisms and novel variants. But it's more, I guess more specifically to sort of analyse gene-sized sequence, which isn't yet possible with individual reads. So the sort of the background, the genomic assembly, this is the sort of basic approach we have for genomic assembly, where we have a single genome, and we make a whole load of DNA fragments, and we sequence these usually with paired end reads, occasionally with single end reads, and we end up with a whole bunch of reads which we assemble into contigs and then scaffold to a nice beautiful assembly at the end of it. And this is all enabled with sequence graphs, and for next-gen reads, the sort of most common approach is the De Bruijn graph. And a De Bruijn graph is a graph of connected k-mers, and neighbouring k-mers overlap in all but one base, and edges give the transition from one k-mer to the next, the nucleotide that needs to be added to one k-mer to move on to the next, to convert to the next k-mer. So very quickly, how we assemble these De Bruijn graphs: we take sets of reads, and we make k-mers from them. So we scan a sliding window over the reads, generating k-mers as we go, and these k-mers become nodes in the graph. And so here the circles and the nodes, and the number indicates the coverage of that k-mer, how many times we've seen that k-mer in the reads. So in this example, we've converted one read into k-mers, and we move on to the next read, and we generate new k-mers where they're not in the graph. And then when we get to the point where we find a k-mer that's already in the graph, then we update the coverage. And in this example, this has enabled us to join together the sort of two separate subgraphs from the two reads. We end up with one very, very simple graph, and we keep going, and the remaining k-mers are all in the graph already, so the coverage gets updated. And so then the idea is that we can build a contig from this very simple graph. We start at an edge node, and we follow edges, adding nucleotides, building k-mers. And this is all very straightforward in this very simple example. If we add a second read, now we've deliberately chosen this read to represent a part of this sequence, but with a SNP or an error at this point. And so when we start adding k-mers from that, we find a lot of the k-mers are in the graph already, but we get to the first k-mer that contains this SNP, and we create a new k-mer for that, and that causes this divergence in the graph. And as we keep going, we're getting new k-mers, and then at a certain point we're able to merge back into the original graph. And so we end up with a graph that that can represent two sequences, really, depending on what you do at this point here. So you can go along this top path and you can output this sequence, or you could go along this bottom path and you could output this sequence. And it's these bifurcations in the graph that are the big problem for assembly. And that was a very simple example. This is still quite a simple example. This is a representation of an E. coli assembly, where the nodes now represent unitigs, sequences of sort of unambiguous sequence, effectively. And the size of the node represents the size of the radius of the node represents the size of the sequence, and the color represents the coverage. And really all this is showing is that even for a pretty simple organism like E. coli, things get very complicated, and there's complicated structures to navigate, and it's a bit hard to see on the screen the links, but basically there's a lot of these kind of structures we've seen saw on the previous slide, where paths diverge and reconverge, and there's examples of repeating patterns as well. And so this sort of leads into some of these sort of common features that we see in De Bruijn graphs, and we've seen these polymorphisms, these bubble-like structures where the path diverges and reconverges. Repeat structures tend to form like this, where you have a number of paths that converge onto a common bit of sequence and then reconverge again. Errors can form these kind of structures, or depending on where they occur in the read, they can form these kind of tip-like structures here, or even join together bits of graph that are kind of unrelated. So genomic assemblers, in order to build contigs, make a number of assumptions. Generally, they're assuming there's one kind of correct genome, and they tend to assume quite a lot about coverage, that there's a relatively even coverage. So this is another simple graph, and genomic assemblers will apply a number of tricks to the graph to try and simplify it further, to make the job of outputting contigs easier. So this graph has now been recolored according to coverage. So we've got areas of low k-mer coverage in red, through to higher coverage in green. And so there's sort of three very common tricks that are employed. They pop bubbles, these kind of structures, and they tend to do that. A common approach is just to take the higher coverage path. So in this example here, we get rid of this lower coverage orange path through the bubble and simplify the graph a little bit. Another trick might be to remove low coverage paths, which are likely to be due to errors. So there's a couple we can identify here and here, which are low coverage paths, so we might get rid of those. And then we might also clip tips, because again these might be due to errors, or at least if they're not adding that much to the graph, because if they're short enough. So again we might get rid of this tip and this tip. And then we're left with a much simpler graph. And then another thing we can do is use paired information to help us to try and resolve repeat structures. And there's an example here. If we had, say, a section of the graph here and here, where k-mers are contributed by two paired reads, then this might give us reason to believe that there's a route through the graph like this, or similarly here we may have a read here that's somehow paired over here, so we can work our way through the graph like that. So that's genomic assembly then. So metagenomic assembly is a different situation. So instead of one genome, we've got multiple genomes. So instead of the one red line across the top, we've got these red, orange, and pink lines illustrating multiple genomes. But the key thing is we don't know what they are. We don't know how many there are necessarily, and we can't. And our DNA fragments are not tied to those. We don't know in advance what they are. So we end up with a whole set of reads that have come from multiple organisms that we've somehow got to assemble into contigs. And so we can't use the kind of standard assumptions that genomic assemblers make, because we've got organisms present at uneven abundance, some very low abundance, some much higher. There can be partial genomes represented in the reads. There are all kinds of biases, enrichment and extraction biases in particular. Similar subspecies can produce multiple branching in the graphs and shared common sequence between them. And there's a real difficulty using paired information in the graphs, because there can be support for multiple paths through the graph and the paired information due to the diverse organisms present. And compared to genomic assembly of similar sized organisms, we've got much higher memory utilization. So what are the solutions? Well, there's really until the sort of last year or two, people tended to use genomic assemblers, and you can get results with genomic assemblers. But really the last couple of years has seen the emergence of a number of metagenomic assemblers. And they're often developments of genomic assemblers, things like MetaVelvet, RayMeta, and IDBA-UD. Vicuna is an assembler specifically targeted at virus populations. But generally a lot of these assemblers are sort of tested or developed with bacterial populations in mind. And most of what we've been doing has been to do with viral populations. And there's also a lack of real comparative data between the assemblers, and it's perhaps hard to know the strengths and weaknesses of the different assemblers. They tend to compare themselves in the publications with the genomic assemblers and less with each other. So we've come up with MetaCortex. So we developed this originally a couple of years ago when we were doing some bat virus work that I'm going to talk about. And at the time there weren't a lot of other assemblers or metagenomic assemblers around. I think MetaIDBA was pretty much the only one that was published at that point. And so we kind of went off on our own route and developed this as well. And we've, as you'll see in a minute, we ended up using it alongside some of the genomic assemblers. But before I come to the bat virus stuff, then the MetaCortex approach, I'll just talk about for a few minutes. So these are the kind of steps we go through. So we have a sort of host filtering step, which is done with a K-based approach. And then we build a De Bruijn graph in a way that is typical for any assembler. We do minimal cleaning on that graph, and then we have an approach where we find all the subgraphs, and then we output consensus contigs for those subgraphs. So I'm going to just talk about those steps in a little bit more detail. So K-mesh filtering, first of all, this is where we have a host library, a host K-mesh library made from a reference. So as I said, we're kind of working a lot with kind of viral populations. So when in the case of the bats, there's a bat host, or we've been looking at sort of viruses in some human samples. So we're the human host we want to get rid of. There's viral enrichment protocols available in the lab which help to a certain extent, but we found they're not necessarily perfect at getting rid of the host. So this is a kind of a simplifying step. So we have a K-mesh library, and we look through the reads, and we make K-meshs by moving this window across, as you saw earlier for assembly, and we compare the K-meshs in the reads with the K-meshs in the library. And if there's more than a threshold value, and often we just use 121mer, often not more than one is enough, then the read doesn't make it into the assembly, basically. So that's K-mesh filtering. We then build the De Bruijn graph, as you've seen before, by making K-meshs from the reads. And there's often high disconnectivity compared to genomic graphs because of all the multiple organisms in there. And then, yeah, we do minimal cleaning, maybe a bit of tip clipping, because again, this in metagenomic data sets, this can still be a useful cleaning approach. So we, in this example, we might get rid of a small tip here. And then we find subgraphs by picking a random node in the wider De Bruijn graph and then growing a subgraph by walking around as far as we can until we get to a point of disconnectivity. Now at the moment our measure we're using is total disconnection from the other subgraphs, but other measures are possible. So we mark all those nodes as visited, and we pick another random node, and we grow another subgraph, and we pick another random node, and we build another subgraph. Having found the subgraphs, we then output consensus contigs for each subgraph. And here we do start to use the coverage again. So we mark the coverage of every node, and we pick the highest coverage node in the subgraph, and then we walk in either direction, following the highest coverage path. So wherever there's a bifurcation, we follow the highest coverage path, and we grow this path through the graph. And then we output a contig which represents that path, and that's our consensus contig for that subgraph. And we then remove that from the graph. So in the original version of this, that was all we did for each subgraph. We just removed that contig. We now remove that contig and then sort of look at the leftovers as well. And so if there's any big enough subgraphs left over, we'll walk those as well. So this graph's too small, so we might remove that. But then this graph might be big enough, so we'll output another consensus contig from here, again taking the highest coverage node and then walking a path through it, and then outputting a contig and clipping that. And then this final subgraph is maybe too small as well. So that's the approach in summary. What I wanted to move on to was an example of where we use the tool, and this is in these straw-coloured fruit bats. So they're from a project was carried out in Ghana. And these bats are ideal viral hosts, ideal hosts of zoonotic viruses that can infect humans. And something like 75% of the viruses that infect humans are apparently zoonotic viruses. And these are ideal viral hosts in the location they're from, because they're widely distributed, they're migratory, and they roost very closely together, able to pass viruses amongst themselves very easily. And there's quite high rates of human contact. There was one group in the study that roosted above a hospital, and they're also eaten as bushmeat, and they're reckoned to be a real problem for disease spread in Africa. So the aim of the experiment was to characterise the viruses that these bats were carrying around, to look for novelty and diversity and the likelihood of spillover to the human population, and hopefully contribute towards being able to better plan for outbreaks and understand likely outbreaks. So experimental work was done and 3 sets of samples were collected. So there were plastic sheets put under the roost and urine samples collected. Bats were captured and their throats were swabbed. And then there were some lung post-mortem samples taken. And this was done a couple of years ago, so the reads are relatively short. They're GA2 76 base pair reads. So our assembly approach, as I say, at the time, it was really sort of metagenomic assembly was starting to emerge. So we used 2 genomic assemblers, Abyss and Velvet, and then we used MetaIDBA and MetaCortex, which we sort of just developed for this project. And the approach was to consolidate the contigs from the 4 assemblers. So we did pairwise comparisons and where 2 assemblers output the same bit of sequence, we took the one that was longest. And then these sequences were blasted, initially blast N, but then eventually T-blast X against the NCBI-NT database, a viral subset of the NT database, basically. And there are a number of interesting findings. So eventually these were sort of curated down to 1,300 odd contigs that represented viruses that infected invertebrates, rather. And there were some interesting findings from the virology point of view, which I'm not going to talk about too much now, but there were some sort of novel viruses identified that were interesting to them. And then where particularly novel things were found, these were then confirmed with PCR, so using the contigs to design primers. What we were interested in really was the performance of the assemblers, the ability to identify the different viruses. And so this is some sort of work that was done around the same time, but this is showing only blast M results for 3 of the assemblers. So I've not included MetaIDBA here because we did have some issues with our data and MetaIDBA running MetaIDBA in the manner it's supposed to be run in, so we didn't think it was fair to run it. We've since used the follow-up to MetaIDBA, IDBA UD, which I'll show you some results from in a bit. But so these are just Abyss and Velvet versus MetaCortex. But certainly in terms of the number of viruses that were identified, we were kind of pleased with this, that the approach was identifying a lot more of the viruses than Abyss and Velvet, in around twice as much, sometimes as much as 4 times as many. So this was good. In terms of the length of those contigs, the mean for MetaCortex is not a lot more than the average read size. Than the read size, and this is because it's outputting a lot of very small contigs, outputting far more contigs than the others, but a lot of them are very small. So if we kind of get rid of those small contigs, we still find there's some useful sized contigs there, and our largest contigs are sort of comparable to what Abyss and Velvet were producing. So we were kind of found that reasonably promising. So since that work, we've then rerun the same data set with some of the new assemblers that have sort of emerged in the time since that work was started. And so this is, first of all, some results. Just this slide is just MetaCortex. So this is showing for different k-mer sizes. This is the number of contigs greater than or equal to 100 base pairs. This is the number of contigs greater than equal to 500 base pairs. And then this is the number of good viral contigs. So good defined as a blast N with a e-value less than 0.0001 and a contig length greater than a read length. And then this is the number of viruses, the number of unique viruses in those contigs. So I mean, the first thing to note is that increasing k-mer size gives us shorter contigs, but equally increasing k-mer size has given us more detection value. So there's this trade-off between the length of the contig and the ability to detect the different viruses that are present. And this is seen also, if I move on to the next slide, where we've added in the other assemblers. So IDBA UD and Vicuna have a k-mer setting, so they're shown as straight lines in these graphs. But there's the same sort of trade-off is seen in the different assemblers, really. That so for example, IDBA UD is giving us quite a lot of nice length contigs, but we're not seeing the same maybe as much viral diversity as with, say, MetaCortex, and the same kind of trade-offs are there with the other assemblers as well. So I think well, one thing to emphasise is the warning at the top. This is just one of the BAT data sets. So how much you can conclude from one data set is one thing. But I think it probably does illustrate a truth that there are different tools for different jobs, and one of the difficult things is working out which tool to use for your job and what settings to use for it, and depending on what you do, you'll get potentially pretty different results. So the summary really is that it's hard. I guess as we've heard in some of the earlier talks, assembly is hard, and particularly metagenomic assembly is very hard. And I don't think there's one general purpose tool out there. I think there's space for a few assembly tools and a few different approaches. And it would be good to have better understanding maybe in the field about what's the best approach for particular situations. Longer reads are going to really change things. Again, we sort of heard a bit about that, I think, in one or two of the other talks. Things like the MySeq 2x300 reads are so much more of an improvement over the 76 base pair reads used for the BAT data, and whether things like the nanopore are going to change this field, we'll have to see. But until we've got those really long reads, I think there's still work to do in metagenomic assembly. So we want to keep developing MetaCortex. It's not something we've had a lot of time to do, but we want to understand what it's best at and where its strengths and weaknesses are compared to other assemblers. We want to look at issues like subgraph connectivity, whether our sort of naive total disconnection approach can be improved upon, whether there's more things we can do with graph filtering. I mean, things like Titus Brown has his digital normalization that he's applied to metagenomic data in order to sort of simplify the problem a bit and reduce the read set effectively, and whether that can help. And to look at maybe more sophisticated algorithms for walking the subgraphs. That's another thing I think we want to look at. So just wanted to acknowledge all the people that have been involved in this. So Cortex originally was a development of Mario Kakamo and Zamek Bell, and Mario in particular has been involved in sort of the MetaCortex approach. And then Ricardo at TGAC's also been involved with sort of ongoing Cortex development and a bit with MetaCortex. And then the BATs were a project of Pablo Murcia, who's now at the Centre for Virus Research in Glasgow, and his then PhD student Kate Baker, who's now at the Sanger, and she went off to collect all the samples. And then Mark Alston at TGAC was also involved in the bioinformatics. So that was it for now. Thank you for listening.
We do have time for questions.
Hi.
So I have a question about when you're saying you find more viral diversity in some of the assemblies. How much of that is real, and how much is just that you've separated perhaps the virus into context?
Yeah, I mean, so this is done. So it's measured through sort of getting good blast hits. So they're kind of it's the number, and there's one of the graphs is showing the number of sort of unique viruses. So it's not as simple as one virus being separated into two contexts, I guess. But yeah, there could be other things at work. I mean, we haven't tested with the other assemblers, with the original combined approach where we did Abyss Velvet and MetaCortex and MetaIDBA. The interesting contigs, as I say, were then sort of primers were designed, and they were PCR'd to check that they were real. But I can't remember off the top of my head what proportion of them were actually sequenced like that. But it's a relatively small subset, I guess. So I think that's a fair question. Yeah.
Ollie?
So I had two questions. The first one is, I guess the PacBio sequencer could actually sequence right through the entire length of the virus, but maybe not with high quality. But that would at least give you a sort of template that you could map everything to, and would probably help with some of the ambiguities.
Yeah, I mean, I guess the trouble is with the longer PacBio reads being such such high error rates that the circular consensus reads are obviously a lot less errored, but yes, I mean whether I've not done any work with PacBio metagenomic data, so I don't know. I think that's, from what I understand, it's sort of something that people are starting to do, but I'm not sure it's very well developed yet. I'd be interested to hear if other people have used PacBios for metagenomics, but I think I mean, you know, for something like viruses where you've got such so much mutation and so sort of closely related as well, that having that error rate that the longer PacBio reads have, I think could be a real problem, but it's not something I've done, so I'd be interested to hear what other people have found.
You have another question?
I had another one, I
Go
thought.
ahead.
Okay.
I'll
Do you want to
see
go to somebody else first then?
Another question from the audience? Go ahead, Wally.
Okay. Well, I don't know much about De Bruijn graphs, but it seems, you know, if you've got sequencing errors, you're going to get these little bubbles, I guess, and that's the reason you want to prune them out. On the other hand, if you've got sort of clonal heterogeneity, you're going to get slightly different patterns of departure. So they won't necessarily look like bubbles, but I think possibly there's a danger that if one doesn't actually have something specific in the business of building the graph in the first place, that these two sources of errors versus clonal heterogeneity could actually be confused, and you may be getting rid of one when you really want to get rid of the other. So I mean, so it's really a question: are there versions of De Bruijn graphs which actually don't commit you to putting out absolutely every sort of deviation on the graph? Can't you simply say, okay, this one looks similar enough to that one that I would kind of clump those things together, and maybe later on deal with the possibility that, I mean, if you saw, for example, that with a clonal variant, you'd have a number of things which look incredibly similar, which you could sort of simply map to the same part of the graph, and then later on you say, okay, we've got enough of these that we can sort of break that apart. Instead of sort of trying to break things apart to start with and then get rid of the bits that seem to look funny, why not sort of lump them together in the first place and then sort of disambiguate with greater refinement until you get what you want out of it. Do people do that?
Yeah, I mean certainly for genomic assembly, that's there's tools to do those kind of things. So I mean, Cortex, one version of Cortex VAR, is a sort of variant detecting version of Cortex that Zam Iqbal is behind, and he was going to, I mean, I guess he wasn't going to talk about that, but he was going to talk later on today, and I'm not sure he's able to now, but certainly that's one tool that's used for that kind of thing, and there are other tools around. I guess for sort of so it sort of depends what you're trying to do with the assembly. I guess my example is more sort of de novo assembly, where you want to produce some sort of basic reference. But yeah, I mean, I guess what they tend to rely upon is that, you know, if it's I mean, it depends. The bubble can be due to heterozygosity, or it can be an error. And if it's an error, then it's very easy to tell, because it'll be very low coverage because of if it's a Illumina data because of the Illumina error profile. So the sort of coverage-based approaches get rid of in genomic data, get rid of the things that are more likely to be sequencing errors, and there's sort of less of an issue there. But if you're doing something where you're specifically interested in the variants, then you might do a different approach. Yeah.
Are there any more questions? Okay, well, thank you.
Thanks.